Okay, thanks, Gabriella. Thank you all. As Gabriella said, Hopkins is kind of home to me because I basically did my Ph.D. and my postdoc training here, and so even just flying into BWI, driving into the city, there's really this overwhelming nostalgia, and so I can say once you leave here, a little bit of Baltimore goes with you, which may not be a good thing, but we'll see. Okay, so let me assure you I will not go long today because I understand that talks that go long are not all that pleasant. And let me thank you in advance for your attention. So as Gabriella said, my lab and my research has focused on somatic sensation, so how we perceive information by touch, and also how that contributes to the way that we can interact with the world. And so today I'm going to be telling you a little bit about sort of a high-level overview of touch, how we can behave, and maybe some things that you may not know about, things that could affect touch, and then hopefully the big picture is how can maybe we all sort of incorporate this into the way that we think about intervening for patients who have sensorimotor deficits or just sort of high-level type of take-homes from this. Okay. So before we get into the details, again, I'm saying that why is touch important? Right? So I think everyone recognizes that touch is important because you want to use that to perceive your environment. Right? We're sensing the environment. We sense things that are touching us. We sense things that we're touching. So this is my daughter being born, and immediately she's touching my hand, and I can have this sort of immediate contact. So there's a sense of even social communication. And what I also want to make the point of is that without touch, we would really have a hard time interacting with the world, right? So it's sort of really critical for guiding and shaping our interactions with our environment through tools, through our limbs. My research has looked at basically different aspects of touch, including how we combine somatosensory information over the body, over our hands, how we have other sensory modalities like vision and hearing that may shape the way you feel your touch, and then trying to understand how are the parts of your nervous system that are dedicated to perceiving information on your body, how are they shaped, how are they patterned, how might they change? And just a very important note that I have no disclosures to declare, and I am not a rich person in any way. Okay, so again, I think I need to state some objectives. So the objectives from today will be that by the end of my hopefully engaging talk, we'll all be able to recognize how the brain is organized to support sensorimotor functions. Hopefully everyone can identify how sensorimotor processing is then modulated by things like attention and multisensory influences. And again, this is where I want to already sort of put as, not a challenge, but for you to think about in all of your work, how might these principles apply in some way? And then I'll end with some work that we've done recently with amputees to see how does the nervous system maybe change or perhaps stay the same in people who've lost their limbs. And I would say we can keep this casual, so throughout the talk, if there's something that you have a question about, please feel free to interrupt. Okay, and now I'm going to lead with the take-home message, so if anyone wants to sort of black out for the next 30 minutes, you can do so. Take-home message is one, successful and dexterous behavior requires sensorimotor control networks that can be more distributed and possibly more complex than what our textbooks tell us, right? So the textbook, as I'm going to go through, you have sensorimotor cortex up here, sort of straight up above your ears, right? And sort of your right part of your body is in the left hemisphere and vice versa. I'm going to talk about some evidence that supports that, but also some evidence that says it's a little bit more complicated than that. Second part, which is, despite the fact that the brain is really organized in this really modular way, right? Again, you learn in your textbooks, you have sensorimotor cortex here, you have visual cortex back here, you have auditory cortex down here. In fact, the brain is much more interconnected in a functional manner, and so it almost doesn't make sense, in my mind, to think about these different sensory domains as unique and independent. And then lastly, I'm going to emphasize how the way that we perceive and process sensorimotor information can be very adaptable, although also sort of, maybe weirdly, rigidly patterned too. And I recognize that that seems like a self-contradiction, so I'll sort of clarify what I mean by that. Okay. And again, my goal and my hope is that y'all can sort of think about this. I should also say I moved to Texas 6 years ago, I grew up in the East Coast my entire life, and then now I say y'all. But let me also point out that y'all is a very efficient use of words and also very gender neutral, so I feel like it's a good thing. Okay. Why is somatic sensation important, right? This is one of those things where a bunch of people who are interested in somatic sensation are walking in now, so clearly it's important. This is something that actually when I lecture medical students, I sort of start with this example, right? Because I think everyone in the room can maybe appreciate what happens if you lose your sense of vision, right? If you're blind. Or maybe everyone has a sense of what happens if you lose your sense of hearing. But I don't know how many people actually know what it means to lose your sense of touch. So I want to give everyone sort of a clear example of this. So on this video on the left, you see a woman who's got a very simple job. Her task is to just pick up a match and light it. Okay? And so this is very obvious that she can do this. She can see it. She can do it very easily. Now on the right, you have the same exact woman who now you already know can do this, but the only difference is her hand has been anesthetized by a local nerve block. So she has no sensory information coming in through her hands. And what you can clearly see is, despite the fact that she demonstrated expertise on this, she has sight of her hand so she can use vision to guide this. She could barely pick up the match, she can barely position it properly and hold it tightly in her hand. And now you're like, please light this, don't burn yourself. Got it. Right. And so what I want to emphasize, and hopefully what's very clear, is that without sensory information, even if you have other sensory modalities to guide your interactions, even if you have fine dexterity of your hands, you really lose the ability to have coordinated behaviors. Okay. And so beyond just this very artificial situation where you have your hand magically anesthetized, why else is this important? Well, as technology is evolving, as people are working on things like brain-machine interfaces, right? This is where the idea of understanding what the principles that underlie touch are important, because we want to be able to incorporate sensory feedback into these brain-machine interfaces. So here is a patient who is a quadriplegic. So he is sitting in his powered wheelchair here. He's controlling this robot arm with electrodes that are implanted into his sensorimotor cortex, right? So that by itself is pretty amazing, right? This is science fiction-y already, that he can just sort of think about doing this, and then he can reach over, pick this thing up, drop it over. But what I also want to point out, sort of moving beyond this marvel, is if I were to ask any of you to go and pick up a block and move it over, you'd be like, boom, boom, give me another, boom, boom, where does this go? Let me juggle these, right? And this guy is like, zzz, zzz, and he's got these online computer systems that are also doing a lot of signal processing for him, right? And so the point is, without sensory feedback, while we can do motor things, then we're still doing this in a very labored manner that's a very uncoordinated manner, right? And so understanding touch then allows us to start to build neuroprosthetic devices that can now generate artificial sensations, right? So here is Rob Gaunt from Pittsburgh, who is now stimulating
Index.
Mechanically, and I don't know if you could hear this.
Ring.
Right, so now he's touching this robotic hand,
Index.
and the quadriplegic participant now is able to identify which of the
Middle.
hands on the, which of the fingers on the artificial hand is being touched.
Middle.
Right? Because now there's sensors on the hands that are being
Ring.
touched, that then generates electrical
Ring.
signals that are now being injected into the brain as electrical currents
Index.
to parts of somatosensory cortex.
Index.
Okay.
Ring.
So again, pretty cool stuff.
Ring.
Okay, how do I do this now? Okay. Okay, so that technology works in part because we can exploit what has already been long known and really made famous by Penfield, where we have in sensorimotor cortex what's called this clear body map homunculus, right? And so everyone, I think, understands that in the medial part of the brain, you have your lower limb representations. As you start to wrap around and get more lateral, you now are going to traverse through a large hand representation. You can kind of resolve fingers in that, and then now get into a large face and mouth representation. This is highly conserved across species. This is highly conserved across individuals, although there's a lot of individual differences as well. And this is mirrored both in motor cortex, which is more anterior, and then in somatosensory cortex, which is more posterior. And I already want to make the point that our brains evolved to do stuff in efficient ways, right? And so the fact that, again, your sense of touch is really, really intimately tied into your ability to do stuff, right? Your motor side, it kind of makes sense that the brain would have these two representations right next to each other, mapped in this very systematic and clear analogy, right, which is organized by the body map. And so these brain-machine interfaces at this point to say, I felt touch on one finger or I felt touch on a different finger, it works because we have this organization, the somatotopy that's there. Okay, so we can leverage that. But now, what other information does the brain process for touch? And although I say that, I'm not really going to get into the details of that, other than to sort of highlight some fun little things that we could talk about. Okay, so I think one, again, textbook principle that we know and we teach is that, at least in primary sensory areas, you have this contralateral bias in the representation of the limbs in cortex. So how do we know that that's true? Well, I'm going to tell you a little bit very quickly of a study that my lab has done, which again sort of hits just some fun little tools that we can use and that are also used in the clinic. So I think many of you are likely familiar with transcranial magnetic stimulation. And so the principle here is you have some electromagnet. Once you create this turn on and off this magnet very quickly, you have magnetic flux. And so that flux is going to induce electrical current in cortical tissue that's underneath the coil. And so if you have different arrangements of the coils, you can have different profiles of the of the magnetic field that you're generating. And this is happening very, very fast. Okay, so how can we use this in a research environment, and also how can we use this in a clinical environment? So we can use this, for example, to non-invasively activate and drive activity in populations of neurons. And so if you were to stimulate over primary motor cortex, you would engage in these upper motor neurons. They would then send their information, decussate, go down, synapse onto these lower motor neurons, and then that would drive muscle activity. And so here you can see me applying stimulation to my postdoc's brain. And so you can see he twitched every single time this thing little flashes. He's twitching because now we're sort of directly engaging his hand representation in his brain, causing his muscles to contract. Right? So as much as he would want to resist this, he cannot because we have brain control in my lab. And so in the clinic, this is a useful tool because now you can use this as a non-invasive probe to map what is the function in different parts of the brain. Right? So that's one way. And then what I'm not going to talk about today is the fact that you can also use this type of stimulation and other forms of non-invasive stimulation to create persistent changes in the brain through neuromodulation. Right? So that's something FDA in 2008 approved TMS as intervention for drug-resistant depression, for example. Okay. So we've used this very simply just to ask basic questions, right? So I'm not going to get into the sort of finer grained details of this study that we published a few years ago. But the point of this that I'm talking about today is we can use this to test how contralateral is the representation of the hand in your brain, right? And so we had subjects do a very basic task where they were either going to feel touch on their left hand or on their right hand, on both hands, or on neither hand, right? So they know this. And we say, now, did you feel touch? Where did you feel it? Right? So we call this the Lurban test, right? There's 4 alternative answers. And so without TMS, participants are basically perfect at this. They can reliably tell us when only one hand is touched, where both hands are touched, or when there's no touch. When we applied TMS to parietal cortex on the left side, we found that the behavior would be changed. And what we found was on the left hand, right, so ipsilateral to the side we're stimulating, their performance is still nearly perfect. But on the right hand, now they started to miss touches, right? They would not be able to feel when their hand was being touched, despite the fact that everything was the same. And when they were not responding, they're actually saying nothing happened, right? So this is almost like you're creating this temporary numbness of the hand. They just don't detect this. Similarly, in the bimanual conditions, now they would miss them, but now when they miss, they would actually say, I only felt something in the left hand. So again, it tells us that it's selectively affecting the perception of the right hand when we stimulate over left cortex. So this is consistent with the textbook view, right, that we have, yeah.
So just to clarify, so on the motor side, you're eliciting distinct motor
Yeah.
activity, but on sensory, when you do it, there's no, I feel on my
Yeah, right.
thumb, but you can causing something.
That's right. So with the motor side, you're evoking some sort of output. On the sensory side, we don't create phantom sensations, right? So there's not sort of an equivalent of sort of a phosphine that we can generate in touch with TMS. Mostly we can just create this almost like an equivalent of a scotoma, but in touch. Yeah. Okay, so we can just quantify, again, this is just showing the point that I made, which is you're selectively disrupting perception on the contralateral hand. And this is just to make the point that this is almost in nearly every one of the 30 subjects that we tested on an individual basis, we can see this effect. So again, this is not some, sleight of hand of averaging over a large group and then showing something. This is on an individual basis, we can see this. Okay, so that's the textbook view. And now what I'm going to transition into, again, in a very sort of high level summary, is some functional MRI experiments that my lab has been running where we want to see what parts of the brain are responding to left hand stimulation, and this is now vibration stimulation on the fingers. So it's left hand or right hand only, or both hands being stimulated simultaneously. Okay, and again, the details of this study I'm not going to get into. So the way that we think about this is basically you have some vibrations that happen on the hand that we're measuring bold signal changes measured with functional MRI, and then now we can say what is the amount of signal change that we see for different left hand conditions, different right hand conditions, or when the both hands being stimulated in unison. So this is just showing an example subject where we get significant modulation of the bold signal in essentially across any of these conditions. On average, there's some kind of change. And what we can see is with vibrations in particular, we're getting a lot of activations in these lateral parietal cortex areas or more posterior parietal, but not really too much in primary sensory cortex. So I'm going to bring that up again a little bit. But again, this is just fMRI to see what parts of the brain respond to touch. And again, we can look at this at an individual subject level to avoid group averaging, and there's pretty consistent patterns across our subjects. So the more interesting question is now, can we take an individual voxel, which is 2 millimeters isotropic of the brain, which again contains, let's say, millions of neurons. So in terms of spatial specificity, we're not really getting into these fine-grained neuroscience questions, but simply want to say, if we have a 2 millimeter chunk of the brain, how much does it respond to either hand alone or both hands together? And so we can quantify that in a very simple way, which is just sort of the average absolute change in the signal. So now each voxel is described by 3 points or 3 values. We can then look at all the different voxels, and you see there's a range of different type of response patterns, right? So some respond a lot to the left hand, some respond a lot to the right, some do combinations of those. Then we can just apply a clustering algorithm, and of this chaos, this structure pops out. And we can see that there's actually some really consistent types of responses that you see. And it turns out that there are some voxels, again, that only respond when the left hand is touched, others that only respond when the right hand is touched, yet others that respond only when both hands are touched together, right? This set of voxels responds to either this hand or this hand, but not when they're together. And then there's this other little group that's when both hands are together touched or just the right, okay? So the point here is that there are these consistent response motifs that can be found in these hemispheres of sensorimotor cortex, all right? Yeah?
Are the decreases meaningful, like in this example?
Right.
Touching both actually decrease the activity?
Great. So yes, they're meaningful. In this talk, I'm not going to get into the details of that, but this afternoon we could talk about that. And so here our index is simply how much does the signal change by touch, regardless of the direction. Okay. So I think the important question that I want to highlight now, and really everyone should be thinking, is these different types of classes of responses, how are they distributed over the brain, right? Do we see this lateral bias or contralateral bias as one would suspect? And it turns out we don't with this data, right? So if we look at this, we actually see that these different classes are sort of scattered in what I'll call more of a salt and pepper type of organization, right? It's not that we see left hemisphere primarily responds to the right hand and that right hemisphere primarily responds to the left hand. All of these different types of responses are really mixed up in lateral parietal cortex, right? And so again, this is the point that I want to make, that depending on the way that we think about our data, maybe because we're thinking about the negative changes in the response in addition to the positive changes, then that shows that the brain is actually responding in these more complex ways that are less organized in the simple contralateral bias that we typically expect. Okay? Okay, so that was the first part. Everyone take a little breath. I'm going to take a little breath and take a sip. And I think the rest of this is going to hopefully be a little bit less data heavy. I don't know, maybe not. Okay. So, right. So the second part then is about modularity, right? And this is, again, moving away from just touch and motor behavior and really think about what else influences the way that we can behave and sense through touch. So I just talked about sensorimotor representations of the hands. And again, I made the point that motor cortex, sensory cortex, they're all sort of right next to each other straight up here. Visual is back here, auditory is back here. They seem like they're pretty dissociated from each other. But the point that I want to make in the next part of the slide is that they're really not independent and they're always almost systematically influenced by other senses. I'm not going to get into the details. I'm just going to plug my body of research has shown what you hear will systematically influence the way that you perceive vibrations, right? So if you feel bzzz and you hear doo, that doo is going to change the way you feel the bzzz, and it's going to bias it towards the sound, all right? That's this first part. We've also shown that there are many, many parts of the brain that respond both to sounds and to touch in ways that are very, very specific and reflect this interaction. And then we've also used some of the brain stimulation technologies to show that we can actually stimulate somatosensory cortex and then disrupt auditory perception, the way that you hear sounds. So I'm not going to get into any more of that. But I will highlight that why is this important beyond just sort of interesting little parlor tricks where you get to make sounds in a talk, right? I mean, it's important because to the extent that the brain is integrating touch and hearing information, then again, from a clinical perspective, we might be able to leverage these multisensory relationships. For example, in cochlear implant patients, as you're giving them an implant, the brain doesn't really know how to use that peripheral signal, right? But if you have touch that is also engaging in those same cortical circuits, you can think of touch as sort of training wheels that trains the brain to say, when you hear this, this is what that means, right? So this is one way to think about that. Or you can maybe engage in cortical hearing circuits for tinnitus and now shape the auditory cortex activity using vibrations or accessing the same representations. Okay. What I will talk a little bit more about is just how our vision can influence touch, right? So again, rather than just thinking about how do you do stuff on your hands, but now what are you looking at? How is that potentially going to change, even if it has nothing to do with the behavior that is at hand, literally. Okay. So we had a very simple question that we just want to say, even if it's not about perception on your hand. In fact, let's not even worry about doing anything with the hand. Let's just look at stuff near our hand. How does simply directing your visual attention near your hand change the way that sensorimotor cortex is processing motor stuff, right? The excitability of motor cortex. Okay, so the way that we did this then is we had subjects either have to do a visual task that would require them attending in the space near their hand or attending away from where their hand is, right? Two possible locations. And we basically said in a block of trials, the visual thing is going to happen here, close to your hand. Respond anytime that happens, right? And so most of the time during that, it would happen there 80% of the time, but 20% of the time it would actually happen on the other side, and they would also have to respond, okay? So their job is just to detect when does this visual thing happen. In the second block, then we can tell them attend over here. This is where it's more likely to happen. And again, 80% of the time the light would happen there. 20% of the time it would happen over here, right? So this is the way that we can direct their spatial attention to regions close to their hand or away from their right hand. And we can then confirm that they're doing this because behaviorally the idea would be if they're attending to the location that the light appears, they're going to be faster at responding to that. As opposed to if they're attending here and the light flashes over here, they're going to be able to do that. It's going to be slightly slower, right? So the prediction is the response times is going to be our readout that they're attending where we want them to attend. The more interesting question is now what happens if we TMS over motor cortex and we measure EMG of the FDI muscle on the right hand, right? And the prediction here that we had was if you're attending to this space, you're going to automatically increase cortical excitability to the muscles that are in that space, right? So then the prediction is when you're attending to this hand or this space near the right hand, you're going to have larger MEPs when you TMS motor cortex and you cause that twitch, compared to if you tell them attend to this side and we TMS motor cortex, they're going to have a smaller MEP that we measure, right? And again, I want to emphasize that their task has nothing to do with the hands. Simply the hand is there, and it's just how does attention happen to do something near it. So what do we show? In nearly every single subject, we confirm that when you are attending towards the hand, that the light appears, or the side that you're appearing, you respond faster than when the light appears on the other side. Right? So this is the first prediction. this proves that subjects are attending where we are telling them to attend. And what we also see then is when you're tending to the space near the right hand, when you TMS motor cortex in the left hemisphere, you get a larger muscle activity than when you TMS motor cortex and you're tending away from this hand. Right? So again, this is telling us that they don't need to do anything with the hand, but just by simply looking near their hand, that's changing how motor cortex is responding to their hand. Yeah.
How are they responding again? Is it with the left hand or?
Good question. They're responding with foot pedal presses.
Right.
Yeah. So we make sure that their hands are not involved at all. Yeah.
You kind of have a nice within subject control. Do you
That's
do
right.
The other side, or?
So we did not do the right-hand side, but in some sense we figured you could direct attention both sides. We had a follow-up where, actually, we have in some subjects EMG recordings on both sides, so we can see that it's specific to the side still. Yeah. I mean, it's not the contralateral TMS, but we can at least show that baseline EMG and the sort of spontaneous minis are not different. Okay. Let me just skip that last little bit. Okay, and so what this suggests is that, so why is this important? Why should anyone think about this? So one possibility is that simply looking at things and having something flash near your hand, your brain has this reflexive ability to say, I need to do something, right? So then you're already preparing your brain in some automatic way to say, visual information that happens near my hand dynamically is going to require some sort of behavioral intervention on my side, so then my hands are already sort of in a prepared state. Okay. So that's on visual influences on motor. Now this is visual influences on tactile perception. So recall that I described that Lurban task before, right? Did you feel on the left hand, on the right hand, on both hands, or none? Here we titrated the amplitudes a little bit, so performance is at least unbiased without any sort of visual information. But we also played this little trick where we said, you're going to have a little flashy lights on your fingers. Just ignore those, right? They don't matter to you. Only report when you feel touch on your left hand, on your right hand, on both hands, or if you don't feel touch at all. And what we found is, despite that instruction for the participants to ignore light, they were unable to. And so when we crossed this behavior with light that flashed on the left side, all of their tactile performances then became biased towards the left side, right? They're more likely to report left when it appeared on left only, and when it was on both hands, they were now also more likely to report that it was only on the left, right? So this tells us that a little flash of light is going to even change the way that you experience touch on your hands. And so we then could see if we had left a bright light on one hand, a dim light on the other hand, behavior gets biased towards the bright side. When it's bright, it's flashed alone. Behavior gets biased to the dim side when the dim side is flashed alone. When the bright and dim are both flashed, now behavior is biased towards the brighter side, right? So there's actually this competition between the visual cues that are occurring at the same time. If there's a brighter one, there's more salient than the dimmer one, that brighter one captures your behavior more. And so we can quantify that bias by just saying for every single subject, positive numbers means you're biased towards the bright, negative numbers says you're biased to the dim. And we can see that in these different conditions, you see the strength of these biases. All right. And the last little thing that was kind of interesting for us and that I think is a little bit more puzzling, but potentially something to, again, think about in more of a clinic patient rehab setting, which is even the experience of light was able to bias behavior subsequent to that, have offline effects. So what do I mean by that? When we were interleaving these flashy trials, there were also trials where no light was presented. But even on those trials, we found that after people had been exposed to this bright light on one side, their behavior started to be biased towards the brighter side, right? So that tells you that your brain is, again, just almost like a statistical learning machine. And it says, whatever is happening, whatever is important to me, or whatever may be important to me, I'm going to learn that, I'm going to use that and incorporate that into my behaviors. And so the question then is there some way to now think about, again, these multisensory relationships, multisensory functions, and then leverage that as you're trying to now treat maybe things that we normally would think of as more isolated impairments or deficits.
How long did that effect last? Was that like 5
Yeah.
minutes?
Right.
Next day?
Good question. So don't have an answer for you. Only in our block, and we now have studies to see what the sort of unlearning of this might be. Yeah. Okay. So last part that we're going to move into is sort of this adaptability, right, of sensorimotor processing. So what I just actually described on the previous slide is the way that we perceive information, the way that we use our hands can change very quickly, right? So that seems like it's a very adaptable system. And the example was you can recalibrate localization on your hands. But now the question is other things like the somatotopy, right? The sort of body map that you know in your textbook, is that also adaptable? So this is where we're going to ask this question where in amputees or individuals who've lost their limbs, what happens to this homuncular map, right? And so again, here's the body map. If we just take able-bodied individuals who have all of their limbs and we say, as we're scanning them with MRI, we say, just move your left ankle, right? So we tell them sort of rotate. You can't see this, so I'm not going to demonstrate, but you kind of rotate your ankle, right? This is the consistent part of the brain that is active with fMRI that we can measure, which is corresponding to this lower limb representation, right? So now, a number of things can happen that would lead to amputation of the lower limb. So you have some trauma, you could have cancer, you could have diabetes that leads to a cascading series of events that would then ultimately lead to amputation. And so in individuals who are now missing part of their lower limb, what happens to this somatotopic representation? And so again, we can do this with fMRI. So we did a very simple experiment where in different blocks, we just had our participants move different parts of their body. So for these 12 seconds, they would move their left ankle. For these 12 seconds, they would move their right hand, right ankle, lips, left hand, and so on, right? So very simple block design. And with our amputees, it turns out, even if they're missing their limbs, almost every single amputee is able to experience phantom limbs, right? So they still experience as though they had a limb, it's just clearly not there, and they know this, right? And the cool thing is you can actually control your phantom limb, right? So we can ask our amputees during this block, when it says left ankle, rotate your phantom left ankle, right? And they're like, what? And it's like, yeah, just move your left ankle, control it as though you had it, right? Not just visualize it, but control it. And so that is the instruction. And so when we look at our amputees, now this is the 18 amputees that we tested. And what you can see is, again, in essentially nearly every one of these amputees, when they're moving their phantom ankle, this part of their brain that is part of the lower limb representation continues to be active, right? Some of these amputees lost their legs 47 years prior to our experiment, right? So what that tells us is that the structure that is in the brain is not going away. It's not like you don't have a limb and so the brain is going to reorganize to do other things, right? Here it's clearly still doing stuff that it had been doing many, many, many years before. So we can even look more systematically and say, is there a contralateral bias in the amputees? In fact, we see that when you look in the one hemisphere, there's greater response to the contralateral limb. The other hemisphere, greater response to the contralateral limb. This is true both in the unilateral amputees and in the bilateral amputees. Now the sort of holy grail would be, does this activity in some way predict their phantom pain experience or a chronic pain experience? Because if it does, then we know we should be targeting this in some way, right? Maybe we can go and intervene, change this pattern of activity that might lead to sort of amelioration of the pain. In our hands, the data is not consistent with this idea that sensorimotor cortex responses when they're moving the phantom relates to anything related to phantom pain magnitude or the phantom sensation magnitude or the sort of vividness of the phantom limb movements. It's also not related to depression. It's not related to their ambulation scores measured by the +M. But the only thing that we saw, there was some statistical relationship, was this thing called the SUIs, which is spontaneous use of imagery scale. And so these individuals are the ones that turn out that in sort of other behaviors are better able to sort of visualize things and use their sort of the mental eye, right, to say. And so I think this is somewhat disappointing for us, but such is life, so that the phantom response magnitude then is really not in our hands sort of something that ties directly to this pain response, right? Okay, and so I'm going to wrap up here just by, again, hitting the take-home messages. What I've hopefully conveyed to you today is that successful dexterous behavior really requires this integration of sense and movement of multiple systems, and that could be distributed and more complex than what the textbooks tell you. Also that despite the modularity of the brain organized in different sort of cortical systems, those are highly interactive. And then also, while we can have this sort of very quick fine scale adaptation, there's also evidence that some of the things that are in our brain basically stay that way for possibly ever. And with that, let me just sort of thank my lab from 3 years ago, funding, and then thank you for your attention. Yes.
Thank you for the great talk. I have a question. In your imaging studies, I can see the activation of cerebellum.
Good question. So,
Could you repeat the question?
yes, great question. What happened to the cerebellum? You did not see activations of the cerebellum because the way I was depicting the data was with these surface models of the brain that were inflated. So that's why they did not resemble the brains that you would see normally. And those don't include the cerebellum. We actually do have activations in the cerebellum. They are localized in regions of the cerebellum that are also consistent with a body map. Yeah, and so I just did not show those. Yeah. Yeah.
There's a lot of work showing altered sensory processing in like, neurodevelopmental disorders like autism or ADHD. And in some of the slides that you presented there, you showed that your visuospatial attention modulates how you perceive touch. How much do you think the sensory abnormalities in these disorders is really just pinned down to the fact that you're just being inattentive when they've been tested in these studies?
Right, so the question is, the atypical sensory processes that are observed in neurodevelopmental disorders, how much of that can be attributed to things like attention, right, or just sort of other, so I would say attention, things like working memory capacity, those are all cognitive functions that may not be specific to the perceptual systems, but certainly would impact your ability to judge those. I think that's a great question. That's one that as far as I know, there's not a really clear way to disentangle those, right? Unless you're starting to look at different modalities and that you compare performance across that. And then it turns out that in populations like those with ASD, there could be individuals that show tactile abnormalities, but not auditory or visual and vice versa, right? So then you wind up realizing that in that case, it might not be attention, it might not be these higher order cognitive things, they might be sensory specific. And there are other hypotheses then of what underlies those. So for example, there's recent work that suggests that at a very fundamental sort of neural circuit level, there are operations like divisive normalization that is just maybe not working in the way that it normally does. And so then the way that information is being represented is already being shaped at sort of a population level. Yeah.
So with your laterality studies, have you ever looked at any other body parts besides stimulating the hands? Only because for motor representations, you actually have a different amount of sort of like contralateral dominance depending on the area. So like hands, for example, you have a really strong contralateral bias, but like swallowing muscles, you actually tend to have a much more bilateral representation. So I was wondering if that might follow suit also with the
Yeah.
sensory.
Yeah, so the question is, have we looked at other body parts that may be less lateralized? Not on the sensory stimulation side, but on the motor behavior, right? We had the sort of lip movement to contrast with the hand movements and the ankle movements. I mean, I think it's clear that it's hard to move half of your lips, right? Your right side versus your left. And so I guess that's a short way to say we don't have a good way to look at that. Yeah, because they're often yoked. But I would say that in some sense, the hands ought to be the most lateralized because in some sense we use those most independently. And again, we see evidence for both it's lateralized and it's also highly intermixed. Yep.
So I just wanted to talk a little bit about that point that you just made that, especially for treatment in rehabilitation, we tend to focus on the impaired side alone. But your data and, other data showing bimanual therapies could be very helpful to reinstate the sensory representations and improve sensorimotor integration.
Yeah, I think absolutely right. Thank you for that, because I think that if there's any kind of take home from what I've said, I think that that really articulates that well, which is, again, rather than focusing on only one, limb or organ system or modality, really sort of leveraging the fact that the brain is really functionally interconnected. And so leveraging everything that you can that provides redundant or systematically related information will likely help, I think. Yeah. Yeah.
You mentioned that you can't elicit a perception with like S1 TMS.
Yeah.
So in the motor domain, you can either elicit a movement or you can suppress kind of a tonic contraction. So if you had a constant sensory stimulus against the finger, is there a protocol that you could use to kind of map S1 by having somebody get to the report when they no longer feel
Right.
pressure or touch against the finger? Or when
Yeah. Yeah, so I think that is a good question, and I think that's a good idea. In many ways, I feel like that relates already to the way that we're basically saying when this thing comes on, if we TMS, this is when you stop feeling that, right? So now you're just saying if you have a continuous background, would you actually feel that switch off? My prediction is you would, but it'll be less easy to detect that switch off. But my prediction is that you would. And I should say other people have reported, and I think that this is sort of bad data or at least noisy data, that there are sometimes people who can perceive as though their hands are moving when you TMS S1. One possibility is that in that case, it's actually engaging sort of motor circuits as well, right? I mean, because another dirty little secret is when you TMS a part of the brain, you're activating more than just that part of the brain, right? You're engaging this distributed network. And so this is where I think while TMS is a powerful tool to sort of start messing around with people's brains, it's not such a localized thing.
kinesthesia or proprioception is quite different than.
That's right. I think that that's true too, yep.
Have you done much work trying to improve, I guess, tactile or any other sensory perception with TMS in like a repetitive format?
I have not. So there are some, right, so I mentioned that with neuromodulation, there are some protocols that exist that can change cortical excitability. Other groups have used TMS or TDCS, and now growing to use focused ultrasound, so tying back to the first thing that we talked about today, to actually modulate. Actually, we used TDCS in one study, not over somatosensory cortex, but over visual cortex and auditory cortex, and that showed selective interactions with touch. So, yes.
I think you could shift the threshold.
That's right. So we can improve spatial perception on your fingers by stimulating visual cortex. And so and this is sort of following up on some other stuff with TMS as well. And then if you target a more lateral areas, more over the auditory system, then you can improve sort of vibration perception.
Do you think that's because the auditory area is like it's a timekeeper or some sense, like it processes the temporal characteristics?
Yeah, so the relationship between audition and touch is very similar in sort of the frequency domain of vibrations, right? And so I think that you can think of the auditory cortex as being really good at processing frequency information or spectral information, and then you would be recruiting many of those circuits when you're feeling touch and using that same information, right? Just so people know, the way that you feel textures, right? You hear that ch-ch-ch-ch-ch. That feels different than, right? Because the vibrations that you generate in your skin when you interact with these surfaces are very, very different, right? So what you hear, there's a different pattern of sounds, just like when you move your fingers, there's a different pattern of complex vibrations. And so your brain is basically doing the same type of spectral analysis of those to figure out what is the surface, in the same way that if you hear your voice, it's saying what is the spectral cues for speech, right? Yeah. Yep.
This would kind of suggest that if you were able to somehow stimulate both of those fields, if you will, through different senses, you could really increase the learning
That's right.
capacities
Yeah.
in that way, right? And so you would think that things that had similar vibrations and auditory things could be learned much quicker than things where the sensation and the auditory audition generated by that were more disparate.
That's right.
Is that in fact true?
So we do see that the interaction. So if you have subjects feeling vibrations and judging what the frequency of it is, and then you play sounds, the sounds will bias, again, the way that they perceive those. And the degree to that they bias each other, though, is related in part to how similar the frequencies are. So there is evidence for that.
So you could potentially, like, let's say, when you teach a task where people were learning different
Yes.
textures.
Yes.
If I gave them a stimulus.
Exactly.
That matched that texture, it would theoretically.
Should learn it better.
Improve.
That's right.
Learn it quicker than if I gave them a stimulus that was, if you will, very different, could then be interference learning. Is that in fact known, or is that hypothesized?
So it's hypothesized. It has not been, I would say, demonstrated in such an explicit learning study.
Seems like it would be possible to do that.
Yeah, yeah. And this is exactly the logic that I was describing for how, when with cochlear implants, this is where if you play vibrations that are matching the sounds that the implant should be, representing, then this is a way you sort of leverage that shared representation.
It suggests that people with various types of different impairments, different sensorium, you potentially could help rehabilitate that sensorium by giving them an assist.
That's right.
To a different sense that with a shared overlapping circuitry, where you could then facilitate that recovery or that rehabilitation much quicker.
Perfect. I could not have said that better. That's another take home from this. Yes. Yeah. Yep.
So we saw that there's, I guess, still active parts of the brain that are that can theoretically, I guess, people can use to control their phantom limb. Are there parts of the sensory parts of the brain that we can use to control phantom limb pain? Do you think that
Yeah.
negating that sensory pathway when you TMS?
Right. So, are there representations of the phantoms in sensory cortex that you can access to now affect and modulate pain? Again, that's, I think, one of those holy grail type of things. It's, to date, people have tried to do that. It's not obvious that there is, it's actually not obvious how you engage in a sensory representation of a phantom limb, right, alone, right? And so when we have them move their phantom limb, that's actually getting motor cortex and sensory cortex. It's hard to stimulate a sensory, right? I mean, you can maybe tell them, imagine you're touched on your phantom limb, but that type of imagery, I think, is much weaker than imagine you're moving something. This dynamic kinesthetic thing, I think, is really driving that response. So people are trying to stimulate sensory cortex to modulate chronic pain, phantom pain. I think it's not clear how the evidence is sort of showing the efficacy of that at this point. Yeah.
You couldn't stimulate the peripheral, like the leftover peripheral nerves?
So people are also doing that. Yeah, that's right. Yeah, so I think this gets into the broader question of when you have phantom pain, or if you have chronic pain that's due to some sort of, injury, what is the origin of that pain signal, right? And so you have, pain that can originate from the damage to the nerve itself, or you can have central pain where now there's plasticity in the nociceptive system at the level of the spinal cord, at the level of the brainstem and cortex. So I think it's hard to really, if it were only localized in one part, then you can tap into that. But I think there's so many levels of the neuroaxis that's affected by pain and representing the pain that I don't know that, again, there's sort of mixed results everywhere. Yep.
Did you ask that in the pain questionnaire? Did you ask what type of pain they had? Because I'm curious if it were cramping and tightness, if the movement would relieve it versus.
Right, yeah. That's a good question. We should have asked that. In our offline interviews, we spoke with them once we realized that our questionnaire was kind of useless. And so we could ask things like, how many years have you felt this? What is the scale of this? But it turns out that there's a lot of different experiences of phantom pains, right? And there's actually another thing that some of y'all might find interesting, y'all, again. There's phantom numbness, right? And it's kind of weird that you would be like, you don't have a limb. You're talking about a phantom limb, and yet it feels numb to you. Like, what does that even mean? So that was kind of a cool thing that we realized anecdotally. And then also this other thing that was kind of cool, and again, I don't know how many of you have experienced, or not experienced this, but sort of encountered this. There seems to be a tendency for, in the case of trauma, when someone loses their limb due to trauma, their phantom is oftentimes in the same posture that their limb was in at the time of the trauma, right? So we talked to someone who lost their limb in a boating accident, like the propeller basically hit them in their leg in a way. And so when he experiences his phantom limb, he feels as though his leg is sort of back as though he were swimming still in that posture, right? There's also these anecdotes in some medical books where there's this guy who reports he lost his hand because he was shooting a grenade launcher, so don't play with grenade launchers. And the thing blew up in his hand, and so he lost his hand. But now his phantom is always in the sense of he's holding a trigger, right? So there's this really interesting sort of history of, you know, trauma that's maintained in that representation. Again, all very anecdotal. Okay, yep.
So, in your expertise with the transmagnetic cranial stimulation, what do you think of this company that came out? It's called Halo Neuroscience. It popped up on my social media because of just the Big Brother listening to everything I do, and so it came up as basically these headphones for basically giving an electrical stimulation, and they claim to increase learning ability and precision as well as motor training. So I was just wondering, what do you think of that? Do you think it's just a gimmick? What do you think they're pushing?
Again, notice that I have no conflict of interest declarations. I think that the jury is still out on many, many of those types of devices. I mean, in very controlled research settings, transcranial stimulation is already sort of a mixed bag, right? And so I think to the extent that companies are now trying to leverage that and say, here, buy this thing, it's gonna make you learn something faster or make you a better gamer, right? I mean, this is actually, the gaming industry has sort of really connected with this. I would say I'm not spending money on that, and I'm also not recommending that.
An article about how they gave it to the San Francisco Giants, I think, and then the coach was like, yeah, I made them shave some fraction of a second off of their split time, and as a result of this experiment thing. I think sort of just to echo sort of what Jeff was saying is that a lot of times with these devices, they sort of have this approach of the one size fits all, but really it's a bit more nuanced because everyone's anatomy is different. Half of these resistance of where the currents from the go is a little bit different. So if you can do a better job of sort of controlling that specific current pathway, you can have better success with these just popping on the head.
Well, that's what I'm asking. So with TMS, we're not as honed in as we would like to, but we're pretty accurate. But then with this, it's you're putting on headphones and you're stimulating, you're giving an electrical current. What are you stimulating?
Yeah, I mean, with a lot of these transcranial electrical current stimulations, the idea is 50% of the current is shunted at the scalp already, right? So what is even reaching your brain is not really obvious, and you might feel this little tingling, you're like, yeah, something awesome is happening. I feel so smart. Yeah. Okay. Well, thank you for your great questions, too.